Hi everyone, myself Ishwari. I am pursuing MSc Biotechnology in Sri Krishna Devaraya University in Anandapur. So I would like to explain about DNA damage and repair. So before going to this topic and what is DNA? So DNA means deoxyribonucleic acids and it is simply called as DNA. All living things have DNA within their cells and it is a genetic material and it is a double-stranded helical structure. The structure was proposed by Watson and Crick. We know that most of the DNA is located in the cell nucleus, and but a small amount of DNA can also be found in the mitochondria. So the main role of DNA in the cell is the long-term storage of information. So let us go to the topic. DNA. DNA is relatively stable molecule, but its natural environment is quite toxic, and damage to DNA is inevitable because each cell contains one or two copies of its DNA. So the DNA sequence is highly protected from harm, but DNA can be altered by mistakes made during its own replication or recombination. The DNA damage and sequence alterations to the DNA are often quickly repaired, but when they are not, the DNA becomes permanently altered and harbors mutations. So mutations are changes in DNA sequence, and when mutations occur in germline cells and these changes are inheritable. What is DNA damage? DNA damage is distinctly different from mutations. So although both types of errors are occurred in DNA. DNA damage is abnormal chemical structure of DNA, while a mutation is change in the sequence of standard base pairs. So DNA damage leads to change in the structure of genetic material and prevents the replication mechanism from functioning and performing properly. The DNA damage and mutations have a different biological consequences. Types of DNA damage and damage to DNA includes any deviation from the usual double helix structure. There are six types of DNA damage. First one simple mutation and second one deamination, third one missing bases and fourth one chemical modification of bases and fifth one formation of pyrimidine dimers and sixth one strand breaks in simple mutation, and these mutations are switching of one base for another base, and it occurs in two types, transition and transversion. In transversion in transition, one pyrimidine is substituted by another pyrimidine and purine with another purine. Transversion involves substitution of a pyrimidine by a purine and purine by a pyrimidine, such as thymine by guanine or adenine, and adenine by cytosine or thymine. So other simple mutations are detection, insertion of a single nucleotide or a other simple mutations are detection, insertion of a single nucleotide or a small number of nucleotides. Mutations which changes a single nucleotide are called point mutations. Second one, deamination. So the common alternation of form or damage includes deamination of cytosine and to form a uracil and which base pairs with adenine in nest replication instead of guanine with which the original cytosine would have paired. So as the uracil is not present in DNA, adenine base pairs with the thymine. Therefore, cytosine and guanine pair is replaced by thymine and adenine in nest replication cycle. Similarly, hypoxanthine results from adenine deamination missing bases, and in this the cleavage of n-glycosidic bond between purine and sugar causes the loss of purine base from the DNA, and this is called depurination, and this apurinic site becomes non-coding lesion. And fourth one, chemical modification of bases. In this, any of four bases of DNA leads to modified bases. Methyl groups are added to various bases, and for example, guanine forms 7-methyl guanine and also 3-methyl guanine. Adenine forms 3-methyl adenine, and in a similar way, cytosine forms 5-methyl cytosine. The replacement of amino group by a keto group converts 5-methyl cytosine to thymine. Fifth one, formation of pyrimidine dimers, and it is also called thymine dimers. The formation of thymine dimers is a very common, in which a covalent bond, that is the cyclobutyl ring, is formed between adjacent thymine bases, and this is leads to loss of base pairing with the opposite strand. So a bacteria may have thousands of dimers immediately after exposure to the ultraviolet radiations. And sixth one, strand breaks. So sometimes the phosphodiester bonds break in one strand of DNA helix, and this is caused by various chemicals like peroxidase, radiations, and by enzymes like DNase. This leads to breaks in DNA backbone, and single stranded breaks are more common than double strand breaks. And sometimes these X-rays, electronic beams, and other radiations may cause phosphodiester bonds. These are breaks in both strands which may not be directly opposite to each other, and this is leads to double strand breaks. Some sites on DNA are more susceptible to damage, and these are called hard stops. DNA repair. The repair system plays a significant role in mutation process. As a result of repair, potentially lethal changes in DNA may be eliminated. If the repair system functions in error-free manner, potentially mutagenic lesions are eliminated. So before they can be converted into final mutations. In order to maintain the integrity of information and contained in it, the DNA has a various repair mechanisms. So types of repair mechanisms. So direct or photoreactivation and excision repair system, and forced replicative recombination repair, and mismatch repair. And fifth one, SOS repair. So direct or photoreactivation repair, the ultraviolet light is a physical mutagen and can induce mutations. Ultraviolet radiations causes the formation of pyrimidine dimers. When two pyrimidine bases occur together is a single strand of DNA. So thymine dimer is most common one, but cytosine dimer as well as thymine cytosine may also occur. So thymine dimer is a state in which the two adjacent thymine molecules are chemically joined, so distorting the structure of DNA so that impeding the transcription and replication process. This pyrimidine dimer formation is lethal to the cell unless it is corrected. So a repair mechanism known as photoreactivation can repair the mutations. So when UV radiated population of the bacteria is subsequently exposed to visible light of wavelength of 300 to 450 nanometers, the survival rate is increases. Automatically the frequency of mutation decreases, and this is due to the activation of photoreactivating enzyme photolase, which splits thymine dimer. In the dark, the enzyme binds with the thymine dimer, and in presence of visible light, the enzyme split the thymine dimers. Up to 80% of thymine dimers existing in genome can be photoreactivated. Excision repair. The repair system remove and replace the altered bases from damaged DNA. Excision repair system involves in two ways, that is the base excision repair and nucleotide excision repair. First one, base excision repair. So in this mechanism, modified bases are recognized and cut out. Mutations causes alkylation and deamination of bases, and which are recognized by special DNA glycosylase enzyme. Glycosylase recognizes and remove the damaged bases by hydrolyzing the glycosidic bond and cut out the damaged base, creating AP site, that is a purinic or pyrimidic site. So the AP site is recognized by AP endonucleases, and which splits the phosphodiester bond on DNA strand at AP site and removes the AP sugar. After that, the damaged nucleotide is removed. So the gap is repaired by a DNA polymerase 1 and ligated by DNA ligase. And next one, nucleotide excision repair. So in this mechanism, the defective nucleotide are cut out and replaced. Unlike base excision repair, the enzymes in nucleotide excision repair recognize the distortion in shape of double-stranded DNA structure, so caused by the thymine dimers or intercalating agents. The multi-subunit enzyme excite nucleases, and these are endonucleases and exonucleases activity, and these hydrolyzes two phosphodiester bond, one on either side of distortion caused by lesion, creating 3' hydroxyl group and 5' phosphate group. The excised nucleotide is removed and the resulting gap is filled by DNA polymerase 1 in Escherichia coli and DNA polymerase in E in human, and finally joined by DNA ligase. And in this in base excision repair, one strand has a stretch of damaged DNA. After that, the endonucleases which cleaves the damaged portion by incision. Next, exonucleases integrates the damaged stretch, and this leaves a gap in the damaged strand. After that, the DNA polymerase synthesize, and the new strand of DNA ligase forms. The phosphodiester bonds and which seals the ends. In nucleotide excision repair, in this and two exonucleases and binds to DNA at the site of bulky lesions, in which one cleaves the 5' side and the other cleaves the 3' side of the lesion, and the DNA segment is removed by helicase, and DNA polymerase fills in the gap, and DNA ligase seals the neck. Post replicative recombination repair. So it is a double stranded break repair mechanism. So if a DNA strand contain lesions and which prevent base pairing by creating gap in the daughter strand during replication. This causes break in both strands. So in this repair mechanism, the gap is filled by sequence information from parent strand of sister chromosome by homologous recombination process. The repair of parent strand is done by DNA polymerase 1. The S mismatch repair. In this, sometimes the wrong bases are incorporated during replication process. The guanine and thymine are cytosine and adenine pairs are formed. The wrong base is always incorporated in the daughter strand only. Therefore, in order to distinguish the two strands for the purpose of repair, the adenine bases of the template strand are labeled or tagged by methyl groups. In this way, the newly replication DNA helix is hemimethylated. The excision of wrong bases occur in the non-methylated or daughter strand. SOS repair. In this, the photoreactivation, excision repair, and post-reactive recombination repair are generally error-free repair mechanisms. So, however, there also exist an error-prone and mutations-inducing repair called SOS repair. And the error in both complementary strand of DNA would be lethal for the cell. Thus, SOS repair reconstruct the chemical structure of DNA, but the hereditary information is lost. Hence, SOS repair causes the mutation along with the repair of DNA. So, DNA polymerase 5 help in SOS repair, and it is also known as inducible repair, and it involves more than 40 genes which encodes protein responsible for protection and replication of DNA as well as repair and mutation. The SOS response have been found in E. coli, Salmonella typhimurium, Mycobacteria and tuberculosis, etc., but not in eukaryotic cells. And it is not a single discrete mechanism, but it includes diverse responses and such as the ability to repair thymine dimers and to induce various prophages and to shut off respiration and to delay septum formation during cell division, and these all responses are regulated coordinately. The SOS response is induced when DNA is damaged, or when replication of DNA stops and single-stranded DNA accumulates. In this, the agents such as UV radiations and methyl methane sulfonate, as well as chemicals that damage the DNA, induces the SOS response. So RECA and LEXA genes are the major in SOS response induction. So LEXA encodes LEXA protein, which is repressor, and it binds to SOS box and near to the promoter of SOS gene and prevents the gene expression. And this and RECA protein is a nucleoprotein and it is always high in bacterial cell, and it forms nucleoprotein filaments on a single stranded DNA and protects further damage of DNA. So RECA protein acquires proteases activities and activates self cleavage of lex A protein from SOS box. Now the operator and promoter side becomes free and facilitates gene expressions of SOS box gene. Thank you.